Hi, I'm Janine Cody, and I'm presenting the 2017 research update from the Chromosome 18 Clinical Research Center. And this year I thought I'd do it a little different. Normally I talk about the projects we're working on, what our goals are, sort of the bigger question sort of things. And this year I thought I would focus on what specific people really do and what are the activities that keep people busy every day. And all of that, of course, is in service of the bigger goals and projects, but I think it helps people understand a little bit better of what really goes on a day-to-day basis. So let me share that with you today. First of all, I can't do any discussion about what goes on without reinforcing the idea that all of this happens with a very focused goal in mind, and that is to make the chromosome 18 abnormalities the first treatable chromosome abnormalities. And by that we mean healthy and independent lives for people with chromosome 18 abnormalities. And of course that means everything we do really all culminates with the development and the evaluation of treatments. So those are the things we're working toward and working on. And by treatments, I want to make sure that people understand when we say treatment, we're talking in the broadest sense of the term. We're talking about not just drugs, but foods, supplements, foods to avoid, experiences, like various therapies that might be beneficial that are very specific for the issues that people with chromosome 18 abnormalities might have. So we're talking about all kinds of potential routes, mechanisms, tasks that might be treatment and might be valuable for someone with a chromosome 18 abnormality. And what we do really focuses on three general big categories of activities. And so I thought I would present this focusing on those three big areas. And they are understanding the people, understanding the genetics, and then communicating our results. So, understanding the people. Excuse me. We had to really begin all of this with comprehensive clinical assessments because these syndromes were very poorly described. So much of the literature that existed before we started focused on things like they had low-set ears, but they failed to mention whether the ears worked or not. And if they did or didn't work, how did they work? If they had hearing impairment, what kind of hearing impairment? That information just didn't really exist. The functional information, the information that really makes a difference. So we've done lots of comprehensive clinical assessments, and we continue to do them on different groups of people with chromosome 18 abnormalities in which we haven't done comprehensive clinical assessments in the past. And so those clinical assessments include such things as just clinical exams, endocrinology, neurology, immunology, those sort of things. And they include various evaluations, so x-rays, blood tests, that kind of thing. And these, of course, are tailored to the specific condition that we're assessing at the time. So they might be a little bit different from person to person. But they involve lots of different pediatric and adult subspecialists who volunteer their time for us. We also do a lot of developmental progress sort of evaluations to see how people are developing and progressing in life and how they're growing up. Are they meeting milestones, and what are their strengths and their weaknesses that might need further help and encouragement. We also do psychology evaluations as well as psychiatry evaluations. The comprehensive assessment also includes evaluations by ENT, audiology, and ophthalmology. So those are specialized sort of assessments as opposed to a physical exam. And we also do brain imaging with our Research Imaging Institute. And for adults with chromosome 18 abnormalities, that we work with our GATE lab to look at gross motor skills, and assess how they walk, what their posture is, and try to understand their more physical development. So that's all a part of comprehensive assessment. And as I said, we tend to do those now in more focused groups of people. But we're still doing comprehensive assessments when we have the funding to do that. And that's our biggest obstacle, is the funding to do that. We also spend a lot of time on natural history. So natural history is really understanding what happens to someone through their lifetime. So we actually know, in relative terms, a lot about children with chromosome 18 abnormalities. We know less about young adults with chromosome 18 abnormalities. And so natural history is really learning about what happens to someone across their lifetime. How well do they do in school? What kind of jobs do they have? How do they progress in life? Do they live independently? What supports do they need if they don't live independently? What do they achieve in their life, and how do they live a successful life overall? And then what happens as they get older, medically speaking? Do they have common adult onset conditions? What special aging or typical aging problems do they have? We don't have any idea at this point. And what is their cause of death? Is there something in common that different chromosome 18 conditions have? We don't know any of that yet. And so that's what natural history is, following people throughout their lifetime. And so when people ask me about how long is the study if I enroll, I said, well, it's a lifetime longitudinal study. We're trying to learn everything we can about people with chromosome 18 abnormalities from the moment they are born until they die. And so that we hope and anticipate that these are long lives. So we'll be around doing this for a very long time and continuing to get information that we can pass on to families. And doing all that work, which is, a lot of work to follow over, at this point, 650 people with chromosome 18 abnormalities, and we're enrolling more people all the time. That work is primarily done by this group of people. Bridget Swallow is a psychometrist, which is the same as a school psychologist, but doesn't work in the school. So she's got a master's degree in school psychology, and so she does lots of surveys, and she's the one who really asks lots of questions about how our young adults are doing. She also has a medication survey that she tries to keep up and see what medications people are on, which we found is a really good window to understand what the medical conditions are. Mimi Hasse has a couple roles in the group. When families are here for the comprehensive clinical assessment, she accompanies them to all the appointments to make sure they see the right person in the right place, get parking, don't get their insurance information provided when they shouldn't, that sort of thing. Answer questions of families about what the various appointments entail. But most of her time is spent going through people's medical records. We get medical records by the boxful. And she was trained as a physician in Kosovo, but did not do a residency in the U.S., so she's not licensed to practice here, but she still has all that medical knowledge. And so she goes through everybody's records and abstracts those and gets all the information into our database. And then when somebody emails and says, does anybody else with tetrasomia 18p have this, we can look at our database and see if anybody has ever told us that they've had that. So that's what Mimi does. And Annis Hill is the project manager. So she's the central point for communication in general. She schedules families when they come for visits. She's the one who sends out most of the things to families. And if you have a question for anybody in the team, Annis is the person to email, or at least copy her on the email. Some of us get an awful lot of emails every day, and when the answer takes a little while to look up or to think about or to formulate a long answer, usually some of us tend to wait and answer that later when they've got time to think about it, and then the next thing you know, it's number 57 on your list of emails and you forget. Annis never forgets. So Annis will keep us on task and make sure that whatever investigator needs to answer your question will answer the question. So she has a very big job. And all these people have been with the group for at least 10 years. So hopefully most of you know them and have corresponded with them already. But we also do in-depth assessments, and also the same people tend to orchestrate those sort of things. So in-depth assessments really are the follow-up kind of studies. The things we learn from the comprehensive clinical assessment are often very broad items. So, for example, people with 18q- have delayed myelination of the brain, and the question is, what does that do? And so one test that we have started doing when people are here is the finger tap test. We think this is one good measure of myelination in a functional way. So we've started doing that as just an example. Another example is we did a bone break survey in people with tetrasomy 18p. And from that, we are appreciating that they have bone mineral density problems and are asking them then to suggest to their own doctors they get a DEXA bone density evaluation and then send that information to us. So these are just two examples, but a lot of what we do is understand or identify a clinical problem and then try to understand what are the ramifications of that, or what's the cause of that, what's the root of that condition. And so those are our more in-depth assessments that we do. And then we do what I'm calling reverse characterization, for lack of a better term. All the things we've done for understanding the people so far to this point were all about identifying a problem that someone has, with someone with a chromosome 18 abnormality, and then trying to understand it further. Well, the reverse characterization is once we start to find out about the genes and what the specific genes do, and ones that might just cause a problem when you have one copy instead of two, or 3 or 4 copies instead of 2, then we need to look specifically for the things that we think might be the result of having an abnormal copy number of that specific gene. So just a couple examples of that are the SMCHD1 gene, which is on 18p, and it causes facioscapulohumeral dystrophy type 2. Took me a while to learn how to say that, but only causes that when there's a certain condition also on chromosome 4. So we wanted to know if anyone with 18p- had this condition, had first signs of this condition, because it's adult onset. And so we did a survey first, and then we followed that with neuro exams. And we are still interested in this question, and you'll see further questions and evaluations in the future regarding this. At this time, we think only a small number of people with 18p- are at risk of it's called FSHD2 at this time, but we don't know for sure, and we're still looking, and it may cause something maybe somewhat different when you only have a deletion of this gene. Another example is AFG3L2, and this gene also on 18p causes spinal cerebellar ataxia type 28, and again it's an adult onset condition. So what we did first was to send out a survey to ask if people with the deletion of this gene on 18p had any of the first signs, and we followed that with a neurology exam at the conference. We didn't find any first signs of the typical condition, but one of the aspects of this is dysarthria, so speech that's hard to understand. And we are seeing some of that in our young adults with 18p-. So we are planning speech evaluations, which we will do every year, probably do them every year at the conference, and do them every couple years on each individual to see if, in fact, their speech does change and if this gene is responsible. And once, of course, we find out the gene is responsible, then we go on to next steps about understanding how we can alter the action of that gene and normalize it. So the remote assessments, so many of these things are done remotely. So we're asking people to fill out surveys. We may be asking people to, in the future, wear a Fitbit or some device like that. other mechanisms, or do a video of performing some action, that sort of thing. So remote assessments are orchestrated by David Rupert, who is our technology guru, who helps us really get the technical aspects all figured out. Bridget also is involved when it's a survey, and Annis is a person who will correspond with you about any of these specific inquiries that we have. So that's all understanding the people. It involves a lot of different things, lots of moving parts going on all the time. When it comes to understanding the genetics of the chromosome 18 conditions, let me go over that very briefly. We, of course, have to characterize the chromosome 18 change. So here this shows Patty in the bottom picture. She runs the lab, and so she does the experiments that really define exactly what part of chromosome 18 somebody has a deletion of or a duplication of. And just as a reminder, in the past, before we had molecular tools to do this, if you looked at a chromosome under the microscope, you saw something sort of like the picture across the top. It's an ideogram of chromosome 18, and the red box shows the smallest deletion you could detect under a microscope. But all you could say was, we think there's a missing piece at the end of chromosome 18. In those days, we had no idea exactly what constituted that deletion in that person. Today, we can say that there are, in fact, 17 genes in that region, and the box underneath is an expanded view of what's in the little red box on the chromosome. And you can see the 17 genes on chromosome 18q23 right there, and each gene is depicted by a little vertical line connected by a horizontal line. So some of them have one vertical line, and some have a whole group of them. And you can see them, I'm not sure if my pointer is going to show on this, but for example, here's one, and there's the name right next to it, MBP. So that's the initials for the name, and actually they're kind of shown moving down the page, but in reality it's more like beats on a string, so they're just scattered down the page for readability. But today we can know not only which genes are deleted, but exactly how much of the space in between genes is deleted or duplicated, for that matter. So this is an essential part of understanding the genetics. It's the key and the first thing we need to do. And it helps us identify the key genes, because not all genes cause a problem when you have one copy instead of two, or you have 3 or maybe even 4 copies. So one of the things that we do is keep track of the scientific literature, which there's a lot of, and so there are studies looking for what genes are present in too many or too few copies in a so-called typical healthy population. And so when those are identified, then we can just check those off our list. I'm not going to worry about that, because people who are perfectly healthy have a deletion or duplication of this gene. And so it's not going to be a top priority for us. We also use information from animal models, and by that we mean a mouse that has a deletion of a single gene, let's say, and what those characteristics of that mouse are. If a mouse is missing both copies of one gene on 18p and is able to be born, develop, and be perfectly normal, then let me tell you, that is just not going to be high on my list of priorities of genes I'm going to worry about. We also look at the genetics of the gene and what's known. If a gene causes a condition by a recessive mechanism, so that means that both copies have to be bad in order to have a disease, then I'm not really going to worry about that gene too much. It tells us that one copy is probably just fine. So we use that kind of information that's in the medical literature. But we also use our own information, and this is a little schematic of, in general, the thought process. Don't take this too literally, because this shows 4 chromosomes from people with chromosome 18q deletions. And of course, we're actually using molecular data, not images like this, but this shows you 4 different deletions. The person on top has an interstitial deletion. So you can see they're missing this piece in the middle. This piece at the end is actually attached up here. All the others have 4, or each of the other 3 have a terminal deletion of 18q. And so the people with those conditions have a variety of issues. For example, issue A is found in subject 1, but only subject 1. Subject 2 has condition A, B, and C. Subject 3 has B and C, and subject 4 only has condition number 4. So the way we approach this problem is we say, okay, let's look at condition A. Who has condition A? Only subject 1 and 2 have condition A. So we would define the region that has the gene that causes this is between the end of subject 2's deletion and the beginning of this terminal part of subject 1's deletion. So this region right here has a gene that causes condition A. Likewise, we would do the same thing for conditions B and C. And this would help us identify the regions that are key for the chromosome 18 abnormalities. So taking that information from all of you, from your medical records, from our on-site assessments, we do at least an annual update. And at this point in time, there are 263 genes on chromosome 18, and at this point in time, we think 198 of them, so most of them, really don't cause a problem if you have 1 copy instead of 2. We know less about duplications, but we also think those genes are not so important if you have a duplication of those genes. We think 24 might be risk factors, so that just means for example, someone with autism has a deletion of this gene more often than someone without autism. So it doesn't cause it specifically, but it's one of those, let's keep an eye on that, and it may play a role if other conditions are just right. And there's 22 genes we think are dosage sensitive. So by that we mean that the dose of the genes, which are supposed to be 2, if your gene dosage is 1 instead of 2, or 3 or 4 instead of 2, that those genes potentially cause a problem. And so those are the ones we're focused on. But then there's those 19 you see there at the bottom. Nothing is known about those. They look like a gene, but we don't know if they act like a gene. Nobody knows if they act like a gene. And some of those 19 are in those regions that I showed you, like A, B, and C. Some of those are in those key regions. And so in some cases, we're trying to figure out specifically what those genes do. But this is, all this really takes a lot of time to keep up on all the literature. We're always asking new questions about the various, instead of just A, B, and C, what are certain forms of condition A, B, or C. And so we're asking you more questions about medical conditions, and this is really a culmination of a lot of avenues of information that go into doing this and keeping this up to date all the time, because new information comes about all the time. When we think we have a key gene and we want to know more about it, there's several lines of work that we undertake. One is to look at cells with deletions or duplications of this gene and see how they behave differently. So this shows Patty and Erica in the lab looking at cells and growing cells. And here is an example of an experiment we might do. So this shows the inside of the incubator with 2 flasks, and in this case they have identical cells are in both flasks. And what we can do is use a CRISPR-Cas9 technology to snip out 1 gene from 1 copy of the chromosome, and then ask ourselves, okay, so how do the cells with 1 copy of the gene behave differently than with 2? And we can also differentiate those cells into neurons and other cell types, and ask specifically, how do these cell types work differently when they have a missing or a duplicated gene? And the person organizing and conducting that research is a collaborator, Donna Lehman. So we're working closely with Donna on a couple of emerging projects in this regard. We're also looking at mouse models to help us understand the action of the genes. And likewise, you can use the same technology to delete a gene, a specific gene, one specific gene from a mouse, and compare it to a typical mouse and see how they behave differently. And of course, this work, you don't clip out a gene in the whole mouse. You start with an embryo, and it develops into a mouse. And so I know my pictures are misleading, and sometimes I get questions about that. But in the case of mice, we begin with the embryo and grow them up and then compare the mice with a deletion of one gene or a duplication of one gene to the typical mouse that doesn't have that. And we have a team of people working on that. It's led by Georgiana Gould in the Department of Physiology, and she keeps the mouse colonies and organizes all the testing done on the mice. Jun-Hee Kim is an electrophysiologist, and she looks at how their brains work. And so they've been great collaborators to help us. One of the tasks, just as a way of example, that Dr. Gould's lab does is they look at social behavior. So one of the ways that mice can help us is in tests like this. And so in this test, we have a mouse here in the middle cage, and this cage has 3 compartments, as you can see, but what you can't see very well is there's little doors that connect these 3 rooms. And the 2 rooms on the end have these little cages, and the wire cages on either end. And the cage on the right has a mouse in it. And right in the middle is our test mouse. We want to know how this mouse in the middle, how the behavior is different than typical, if it is different from typical. So a typical mouse, you put them in that middle cage, they look around the middle for a little while. They look left, they look right. Oh, look, there's a stranger mouse. I don't know that mouse. Let me go check them out. So they go off to meet that new mouse. And they get bored with that in a minute, and then they go back to the middle, and they go to the other side, and oh, there's no mouse on this side. So they go back, let me get to know this other mouse better. And so mice, typical mice, like to be with other mice. They like to interact with them. They like to check them out. So a typical mouse will check out the whole cage, but they'll spend most of their time in that room with the other mouse. A mouse that has autistic-like behaviors, they don't really care. They check out every room equally. And so that's one example of a mouse behavior test that can be done, and there's a variety of others. This is just an illustration of a learning test. You put a pellet in one of the arms of this maze. See, we have a mouse in the middle. You set the mouse in the middle, and you just see how you do it over and over again. You do it every day for multiple days, and you always put a pellet of food in one arm, and how long does it take the mouse to learn that the pellet of food is always over here? And so it's a learning test that we can do with mice. And there are lots of kinds of learning tests. This is just one that we can do to understand the genes on chromosome 18. So, taking all that information, then what we need to do is communicate it back to families, because that's the whole point of all of this, is to make sure families understand the information and can use the information to make their lives better. And organizing that is Courtney Siebold, who's our genetic counselor. So she writes much of the information that's in newsletters for the registry and for the research center. She spends a lot of time communicating with families, emailing families, explaining their genetic results to families, explaining the data to families. So that is something that actually takes a lot of time. Actually, all of us help Courtney with that, give her information. I usually call it Courtneyizing. She takes what I write and she makes it understandable to everybody. And that information appears a couple of places. If you don't contact us directly, it's on the Chromosome 18 Registry website under All About Chromosome 18 Conditions. And all those syndrome conditions information was written by Courtney with data from our research center. Likewise, on the Research Center website are management guides for physicians. And again, all the information that comes in for physicians in these guides comes from the research done by our research center. So we're hoping this is a mechanism and written in a way that in a format that doctors will find useful and will help them deliver the best care possible to your children. And again, the staff that makes all that happen are shown here. The one person I haven't introduced so far is Gloria Matthews, who's our administrator, so she keeps track of all the paperwork and the budget. And these people have all been with us at least 10 years. They're really committing their professional lives to you. And so I want you to know that and appreciate that. And they are absolutely committed to you and your children and to making life better for you. So this is a whole team of people. I'm the one you usually see, but it really isn't me. It's really all these people working very hard every single day to make life better for people with chromosome 18 abnormalities, because they believe, like you, that our goal is to make the chromosome 18 abnormalities the first treatable chromosome abnormalities, because after all, they know that it's just biology. So we can do it, we can figure it out, and we will, and we're making progress all the time. And if you have questions, of course, as I said before, ask Annis, because if she can't answer the question, she'll make sure that she sends it to somebody who can answer the question, and she will harass them until they answer it, or at least she will keep harassing them. We try to do all we can to communicate with you, and if you have any questions, please ask, because it's the only way we know what your questions are. We try to guess, but we can't always do that. So please ask questions. Either we need to explain it better, or oftentimes it's something that we didn't know about. And so we need to hear from you, and we look forward to that. So thank you for your attention. Thank you for your support. With your help, together, we're going to make life better for people with chromosome 18 abnormalities. Thank you.